Hi there, I'm Gavin Fish with exploreClarion.com, and this is part 3 in an ongoing investigative series trying to find out the identity of a woman we only know as Penny Doe. Her remains were found by children in Monroe Township in 1990. We know that she was murdered, but we don't know who killed her. We don't even know who she is. So if this is the first part in the series that you're seeing, please go back and take a look at the 2 previous installments. In this part, I'm going to be talking about DNA. Extracting DNA out of 32-year-old bones is pretty tricky business. What I'm learning is that in order for DNA to survive, the right conditions must be present. Now, I talked with a geneticist about a year ago that told me that somebody he knew was able to extract DNA from mammoth fur that was purchased off of eBay. I figured that if DNA could be extracted from an Arctic mammal that's been extinct for some 4,000 years, then getting DNA from 32-year-old remains of a yet-to-be-identified woman wouldn't be that much of a problem. It turns out that I may be wrong about that. But somebody out there knows Penny's real name and probably wonders whatever happened to her. So if I can, I want to use DNA to find that person. But there are several steps that I need to go through in order to make that happen. And as I'm learning, it's actually a long shot. But we're going to take that shot anyway. After my last story on this case, several people reached out to me from different organizations offering to help extract DNA from the bones that Dr. Dirkmaat has been curating at Mercyhurst University all these years. From all the prospects, I selected 2. The first one will be the one that extracts the DNA, and the other will be the one to do the genotyping, and I'll get to that in a minute. So after I selected my 2 volunteers, I began the process of attempting to get a sample of Penny's DNA. Now, I want to point out here that both labs that I chose to work with here asked to remain anonymous. So to get the sample of the DNA, I needed to get a sample of Penny's bones. So I reached out to Clarion County Coroner Dan Shingledecker shortly after I'd made my choice on the labs, and I asked him if he'd be willing to request a sample of Penny's bones from Dr. Dirkmaat. He agreed, and a few days later, I received an email saying he had the sample. Then we worked together to get the sample over to the lab. I must say once again, Dan has been instrumental in helping me as I try to solve this mystery. So thank you, Mr. Shingledecker. Thank you very much. So once the sample was sent to the first lab, I made the long drive from my home in Venango County to the lab to meet with my genetics volunteer to watch them at work. But to my surprise, I was asked to do the extraction myself. Now, I don't want to exaggerate my part in this. I definitely have no idea what I'm doing. I was just the hands that did the work under the watchful eye, under the supervision of a trained technician. Step 1 was to pulverize some of the bone. I have to say, this made me a little uncomfortable knowing that these are the remains of a person, of a young woman. But because bones are porous, it was explained to me DNA would be most likely to be intact inside the bone rather than on the surface. So we took a common cordless drill and dug into the bone, leaving several small holes behind. The bone dust then fell onto a sanitized tray where I collected it with a small metal spatula and put it into 6 small plastic tubes. From there, we added 2 solutions to the tubes, one called a buffer and another which is called proteinase K solution. From there, the tubes went into an incubator for 24 hours, which took the tubes up to 60 degrees Celsius, which is 140 degrees Fahrenheit. This prepared Penny's bone dust for DNA extraction and amplification. The whole process took about 48 hours. So extraction is the process of removing the DNA from the other parts of the cells in Penny's remains. Speaking as a total layperson who really doesn't understand what's happening chemically, basically what happens is that the solution at that temperature, the cells begin to break down. And then there's a process of separating the DNA molecules from the other cell fragments. And once you have that pure DNA separated off, you can move on to the next step, which is amplification. Amplification is the process of making copies of the DNA you extracted. And it just completely fascinates me that this is even possible to do in a lab setting. I mean, our bodies do it all the time. They're designed that way. But the fact that somebody figured out a way to do it on purpose, it just boggles my mind. And that somebody was a chemist named Kary Mullis. He developed a process called polymerase chain reaction, or PCR. He actually won the Nobel Prize for Chemistry for this process. And this is how PCR works. By heating the DNA molecules above 90 degrees Celsius, that's about 194 degrees Fahrenheit, the 2 strands of the molecule, we know that DNA is a double helical kind of a ladder, right? So the 2 strands are separated, they're like split from one another. And then by adding an enzyme to the solution that the DNA is suspended in, one that is present in our bodies all the time, the other side of the strand of molecules is built on to each of the strands that were divided, leaving you with 2 exact copies of the original DNA molecule. And each time the solution is heated up above that 90 degree Celsius mark and then cooled down below it again, the quantity of DNA molecules in the solution doubles. Now, this is a very basic explanation reflecting my understanding of it. But the part that blew my mind is that the enzyme in our bodies that reads the strand of the DNA molecule and then builds on the other side can't actually survive at 90 degrees Celsius. However, organisms that live in the deepest part of the ocean next to like volcanic vents have enzymes that thrive above 90 degrees C. And it turns out that their DNA is replicated in the same way ours is. So scientists use the polymerase enzyme from those organisms to amplify human and other DNA in a lab. Like I said, it's completely mind-boggling. So getting back to that cycle of heating and cooling, right? Each time the solution is heated above 90 and then cooled, that cycle takes about 5 minutes before it's done again. So that means if you started with 1 DNA molecule, at the end of an hour, you would have 2,048 copies of the original molecule. And after 2 hours, you'd have 8,388,608. And that's the process that Penny's DNA went through over the 24 hours that I left the lab. So a couple days later, I reached out to my volunteer to see if we'd been successful in extracting and amplifying Penny's DNA. And their answer was, there wasn't as much as I'd hoped, but I think I've amplified it enough for genotyping. And with that, they sent the DNA off to lab number 2. And from that point on, it's simply a waiting game for me. Now, while we're waiting, we need to understand that and figure out something called genetic genealogy. Genetic genealogy is the practice of finding people who share certain segments of each other's genetic code. So in our case, we want to find people that have segments that are an exact match for segments of Penny Doe's DNA. Now, getting back to that DNA molecule that is arranged in that helical ladder that we've all seen depictions of, but I should say in those depictions, we usually see a small segment. In reality, each strand of our DNA is about 6 feet long and about 1/40,000th of the thickness of a human hair. And I've even read that because of the trillions of cells in our body, if you were to take all the DNA strands in the human body and put them end to end, it would wrap around our solar system several times. It's that long, right? But anyway, the rungs on the ladder are made up of, each rung is made up of a pair of what are called bases. And there are really only 4 bases. There are actually only 4 different bases. They're called guanine, cytosine, adenine, and thymine. And when mapping your DNA, scientists take the first letter of each base and write out the genetic code as a long word spelled out using G, C, A, and T, the first letter of those bases. And when I say it's a long word, I mean it's really long. In the human genome, there are about 3 billion pairs of bases in our DNA. So our genetic code is a word with about 6 billion letters. And in all the human family, we share about 99.9% of the same genetic code. And that doesn't matter who you are, where you're from, what your ancestry is, what your sex is. We all share about 99.9% of the same genetic code. But there are important segments in that code that we know, and by we I mean people who study this, they tend to be different person to person. So what lab number 2 has to do is break up the code into segments and then filter out the 99.9% of the code that we all share, leaving only the places where we differ. But then, because that data set is still too big at about 30 million letters of code, they have to filter it even more. And in the end, they'll produce a report that has between 300,000 and 600,000 letters of code and will send it back to me. And at that point, all of the lab work will be done and I'll be left to do the genetic genealogy, something I've never done before. And this is the process of comparing the report I get back from the second lab with reports from others who've uploaded their genetic information, similar reports, to public DNA databases. And hopefully we can find somebody who has a match for a few of the segments of Penny's DNA, making them a genetic relative. And then it's all about finding the genealogy of that person and tracing it back to a common ancestor of Penny's and working back down the family tree until we find the identity of Penny herself. Now, I love family history research, but I've never done anything like this before. The good news is, though, that I have a couple more volunteers waiting in the wings to help me on that part of my journey. So in the fourth part of this series, I hope to be done with the genetic genealogy research and may even know who Penny Doe is.